Hello everyone, and welcome to this audio podcast. This audio podcast an Erythropoiesis edition. This audio podcast as part of the Hurricane Plexar. This audio podcast as part of the Labonix Vendip. This audio podcast as part of a Storm Robotics production. This audio podcast will as part of this series we're looking to allograft rejection, and look at rejection as a basis in look at rejection as part of and in line to laboratory medicine, pathology, serology, immunohematology, and immunogenetics. So the immune system, or the immune system, the immune system, or in line to immunobiology, defends the body against defends physiology against disease. Lymphocytes are one are one category of white blood cells that assist the body resist infection by identifying harmful foreign substances such as microorganisms and eliminating them. The ability of the vertebrate immune system to differentiate self from non-self largely depends on a group of protein markers. For example, antigens and part of known as part of the Major Histocompatibility Complex, or MHC. These markers are present on the surface of every cell and slightly different in all individuals. And the genes that code for these proteins are found linked together on one chromosome, or chromosome 6, particularly in human immunobiology. And because of the polymorphisms, the varying or the multiple alleles of the human leukocyte antigens and genes, it is complicated to find equal, or has been historically complicated to find equal HLA and MHC from and in context of solid organ transplantation. And if a tissue organ is taken from is from a donor and transplanted to the body of an unrelated host, several of the HLA antigens are likely to be different. And so such an allograft made between members of the same species but different genetics has been referred to and is called a homograft. The host immune system regards the graft or allograft as foreign, and launches an effective immune response called a allograft reject called referred to as allograft rejection. Sort of as a sort of generic terminology. T lymphocytes attack the transplanted tissue and can destroy it within a few days. Before transplants are performed, tissues from a patient and from potential donors must be tissue typed and cross-matched as well as possible. Uh, Allograft rejection is mediated by cell-mediated T cell and humoral immune response mechanisms, or antibodies. Furthermore, the quantity of mismatched alleles also determines the magnitude of the rejection reaction. The more differences, the larger the number of antigens to which the immune response can be produced. Thus, the recipient's immune response could react to, for example, 8 varying donor transplant antigens, even though T cell mediated responses can be generated against the foreign antigens, and the rejection of particular types of allograft may be preferentially mediated more through antibodies than through T cell mediated immune responses or CMI responses, and vice versa. So in general, immune responses against transplantation antigens or transplant antigens mediated by performed antibodies result in rapid rejection, or what's being referred to as hyperacute rejection. At the same time, the emerging understanding of the role of the immune system in transplantation has led to attempts at altering the immune system. Unfortunately, unfortunately, at the same time, there are no effective, for example, regimens at the time to achieve a desired effect. The first attempts are in around where immunosuppression is concerned, involved a total body irradiation. Although irradiation prevented the rejection of the organ, it caused aplasia, which was responsible for a very high mortality rate. Anti-neoplastic drugs such as methotrexate and cyclophosphamide were used for immunosuppression historically, but these agents proved to be too toxic and were associated with poor prognoses or poor results. In the 1960s, in the early 60s or 1960s, prednisone was used to control severe episodes of rejection. Subsequently, 6-merocaptopurine and its successor azathioprine were tested successfully in transplantation experiments in dogs, in canines conducted by an early researcher investigator, Roy Kahn, and were introduced into clinical practices. Equally, azathioprine and steroids were used in combination. Despite some initial successes, the rate of early allograft failure was high in renal transplantation, and only a handful of kidneys function longer than 1 year post transplant. Most of the successful cases involved allografts or grafts from living related donors. So at the same time, in this works there is in this works and in a publication in a 1998 publication, there is an illustration that depicts sort of the MHC, or the Major Histocompatibility Complex. The complex is the MHC complex is used as a basis for all organ donations, and thusly is used extensively before all organ transplants. Recognition of antigen by T cell receptors, TCRs, requires the presence of a second set of molecules, a second set of molecules aligned with MHC and on the surface of antigen presenting cells. The antigen presenting cells internalize large antigens and degrade them into small peptide fragments, and pair up the fragments with MHC molecules, and then export the antigenic peptide MHC complex complexes to surfaces where the MHC complexes are available to interact with T cells, and T cells recognize antigen only when it is associated with an MHC molecule. T cells cannot recognize antigen free in solution like B cells can. And furthermore, whereas B cells recognize the three-dimensional shapes of antigens, T cell receptors recognize primary amino acid sequences. It is HLA region of chromosome 6, and class 1 antigens are present on the surface of most nucleated cells, whilst class 2 antigens are less broadly expressed. DR loci is equivalent to the D locus, but as yet or historically had not been fully investigated. And so with greater differences of MHC complex and HLA characteristics between pediatric recipients, the probability of prolonged functioning of transplant survival had been evidenced as being minimal, and rejection of transplants occurs again mainly through cytotoxic T cell effects. The nephrotoxic aspects and the nephrotoxicity causes more complications, and so at the same time in around around transplant rejection where there had been a mention of hyperacute rejection. There are actually mainly three forms of allograft rejection. There is hyperacute, which is within hours, where rejection is within hours, and the cause is primarily from antibodies, so anti-ABO and anti-HLA. Then there's acute form of allograft rejection, which is weeks to months, which is where the cause is mainly cell mediated, so there's CD8 and CD4 T cells. And then there's chronic rejection, which is months or years, which is where the cause is again cell mediated CD8, CD4 T cells, and antibodies to tissue antigens, and where there's antibodies to tissue antigens. So at the same time, controlling the expression of the antigenic and specific in around the antigen and antigenic responses and immunological pathways. There has been research historically evidence to inform where investigations in around the antigenic responses can lead to the immune system being made tolerant or being on a tolerant basis in context of immunobiology. And so controlling the expression of what's been referred to in literature as ILT3 and ILT4 expression, and where signals from T suppressor T suppressor upregulate expression of ILT3 and ILT4 receptors on the dendritic cells, and ILT3 and ILT4 then signal the nucleus to shut down a production of other cell surface molecules, may allow the immune system to tolerate a donor organ, but leave the rest of the immune response alone, avoiding the risk of infections and malignancy observed with non-specific immunosuppression. Immunologists have been searching for the full explanation of tolerance and around immunological tolerance for the past 3 decades, to help improve the acceptance of transplanted or solid organ transplants. Tolerance prevents the immune system from attacking its own tissues and harmless foreign antigens. Self-tolerance can be partly explained by the destruction of self tissue by T cells and by T cells that react to self antigens, but some self-reactive T cells sort of slip through the destruction process, killing tissue in the body. Convinced an antigen and specific mechanism of tolerance exists, works by a team in Washington and in Rome several years ago, began looking for and then found a different set of T cells. These suppressor T cells induce tolerance after recognizing the antigens on the surface of another type of immune cell called dendritic cells. Dendritic cells which process and present antigens to T cells, including T suppressor and T helper and T killer cells. Somehow, after this recognition, the T suppressor cell robs the dendritic cell's ability to activate T helper and T killer cells to respond to the same antigen. And so, without active T helper and T killer cells, the immune response to the antigen is put on hold. Looking at the outcome of 15 heart solid organ transplant recipients, the team between Washington and Rome several years ago, found 2 proteins that play a role in vivo, and in organ transplant. The recipient's immune system initially reacts against dendritic cells and that come from a donated organ. If these dendritic cells could be suppressed, the patient or the recipient may have a lower risk of rejection or tissue rejection. And so the investigators also had identified 5 out of 6 recipients or patients who had 5 out of 6 patients or recipients who had T suppressor cells capable of suppressing the donor's dendritic cells experienced no acute rejection episodes. 9 other recipients or patients could not suppress the dendritic cells, and all 9 experienced 2 or 3 rejection episodes. Receptor pain may also help in around transplant acceptance. So that draws this audio podcast to a close. This was an audio podcast in around sort of the generics in around looking at rejection as part of this series. This has been an audio podcast as part of the Erythropoiesis edition, an audio podcast as part of the Hurricane Plexar, an audio podcast as part of a Storm Robotics production. Thank you for tuning in.